Welcome again. So today we are discussing on different bacterial exotoxin, and we came to know that exotoxin are the substance that is released outside from the gram-positive and gram-negative, both all bacteria that comes out into the surrounding, that into our body, and then cause the different action. Among that, we have talked about that inhibition of protein synthesis. Then we have talked about we have inhibition of protein synthesis. Then there was like other mechanism of action, like we have talked about increased fluid secretion, that is CAM, then enterotoxicity, E. coli, Bacillus anthracis, Vibrio cholerae, and Bordetella pertussis. Then there was inhibit of release of neurotransmitters, and now also we have talked about lysis of the cell membrane. But there is also another important mechanism by which the bacterial exotoxins acts in our body and cause damage to the body. The main mechanism is known as another one is superantigen causing shock. So there are some bacteria that release the toxin, and that toxin act as a superantigen. Normally, any foreign substance that enter into our cell, enter into our body, act as a foreign, and then antibody mechanism kicks on, and then there will be the formation of antibody. In a normal process, there is only 1% activation of the T cell. Initially, any foreign substance enters into your body, that is picked up by the antigen presenting cell, then it goes to the T cell, into the lymph node, and then activate the T cell. After activating T cell, the T cell get activated, and then it send the signal to the B cell, and the B cell get activated, and then form the plasma cell, and antibodies form. This is the whole mechanism of formation of antibody. In case of normal antigen, there will be only 1% of activation of T cell, but if it is a super antigen, so it doesn't bind into the actual group, and then there will be the bind at the side of the group, and then there will be activation of 20%, more than 20% of this T cell. That leads to the devastating condition inside your own body. You actually, as a rule, there is a provision of activation of only 1% of the T cell, but since the antigen is a super antigen, not a normal antigen, it is activating more, that is more than 20% of your T cell inside your body. And anything more than the normal will going to hamper your own body. And because of that, since the rule was to activate only 1%, but there is a 20%, more than 20% activation of T cell, then there is the many more interleukins, cytokines, and released inside your body, and because of that, your body goes into the shock. Let me show you. So, if you talk about the superantigen are the class of immunostimulatory molecules produced by the bacteria and viruses. Their potent immune effect are due to their unique ability to bind to the major histocompatibility complex outside the antigen binding cleft, and to stimulate the T cell in a T cell receptor via a specific manner. So initially, actually, it should be bind to the major histocompatibility complex into the, you can see in the picture over here. We can show you. Okay, so you can see over here, this is the T cell receptor, and this is the MSC class 2 present in the antigen presenting cell. Anything that has antigen has came will be processed by the antigen presenting cell, and then goes to the lymph node and present to the T cell. Actually, this should be here. There should be this should be bind over here in this part of the region. But by any means, if it is bound to the outside, then there will be the more activation. There will be this super activation of this pathway, and there will be actually 1% T cell should be activated, but there is 20% more T cell has been activated, and because of that, this leads to there's bind outside the antigen binding cleft and to stimulate the T cell. T cell has been activated in a long manner, and because of that, we have found this region, and because of that, there will be this activation leads to non-specific activation of the T cell activation, and that leads to the release of the many cytokines, normally interleukin 1, interleukin C, TNF alpha, interferon, and this will going to have there is a lot of amount of this cystopagogular superantigen had activated a lot of trigger immune reaction within your cell, and that leads to actually a lot of consequences, like it will activating all your immune system, and because of that, you can find that patient develop this toxic shock syndrome, that is the fever, rash, shock, and other toxin cause scalded skin syndrome, and food poisoning. These are not actually the superantigen. We are talking about the superantigen that has the two mainly Staphylococcus aureus and Streptococcus pyogenes, and these two bugs, these two bacteria produce the antigen that is the toxic shock syndrome toxin and erythrogenic toxin of the Streptococcus and toxic shock syndrome toxin of the Staphylococcus. These two have the superantigen capacity, and what they do, they cross-link the beta region of the TCR, that is the T cell receptor, to the MSC class 2 on the antigen presenting cell outside of the antigen binding. This leads to the overwhelming release of the interleukin 1, interleukin 2, interferon gamma, and TNF alpha, and that leads to the shock. So what is happening, that is a huge amount of immune reaction has been triggered inside your body due to this toxin that is released by the Staphylococcus aureus, and the toxin is toxic shock syndrome toxin, and another toxin is erythrogenic toxin A, and that is released by the Streptococcus pyogenes. And these two toxin that is released by the respective bacteria, that is toxic shock syndrome toxin by Staphylococcus aureus and erythrogenic toxin A by Streptococcus species, they are act as a superantigen. They actually, how they activate the immune system, they are binding outside, which I have seen in the figure, and since they are binding outside the group, they are triggering more immune cells. Since more than 20% of the immune cells has been triggered out, there is the overwhelming immune response that leads to the release of the more and more interleukin 1, interleukin 2, INF alpha, INF gamma, and TNF alpha. So interferon gamma and TNF alpha, and this all release inside your body leads to the shock. So Staphylococcus aureus producing toxic shock syndrome toxin that cause the toxic shock syndrome. What are the features of toxic shock syndrome? That is the fever, rash, shock. These are the main component of your toxic shock syndrome features. There are other toxins of Staphylococcus, like toxin cause scalded skin syndrome, that is exfoliative toxin, and food poisoning by the stable enterotoxin. These are the toxins that are released by the Staphylococcus aureus, but they are not superantigen. When in case of the Streptococcus pyogenes, it's really the erythrogenic toxin A, same, this is the since the superantigen, this will be antigen will processed by antigen presenting cell through MSC class 2, bring to the lymph node. In the lymph node, they will go to the T lymphocyte, which has the T cell receptor. They will combine to it, but they are not presenting in the group, so it will bind outside the group, and since it's bind outside the group, there will be again activation of the immune system will trigger will be more enhanced, and since there is more and more production of this chemokines, interleukin 1, interleukin 2, ion interferon gamma, and TNF alpha, that leads to the shock-like feature. So there will be development of Streptococcus pyogenes will develop due to release of erythrogenic toxin A, will develop toxic shock-like syndrome that includes the fever, rash, shock, and scarlet fever. So these are the way by which different toxin, exotoxin, are acting in our body, and they all are equally important in the USMLE point of view. They will help you to form a single, second primary question, or maybe a second or third level of question. So they may give you a, okay, they are talking about the superantigen, or they will mention you that toxic shock syndrome toxin is released from the Staphylococcus aureus, and patient is in shock, or a patient is in shock by a bacteria Staphylococcus aureus, name what are the toxin, or a patient has been in shock with the due to the toxin release, that is a toxic shock syndrome toxin. Now name the bacteria, and you can go through the Staphylococcus aureus. Okay, so you need to, even they can give you one of the differentiation point like Staphylococcus or in Streptococcus. One is catalase positive, another is catalase negative. Even Staph aureus is coagulase positive, so they can give you any information and try to differentiate between these two also. So in this way, we can actually pinpoint what is organism they are going to ask. Thank you.